All right, welcome to everybody trickling in. We'll give people a minute or so just to join before we get started. All right, thank you everybody for joining. Some people are still continuing to join, but let's go ahead and get started. So this is the continuation of the most popular part of our RCN on wastewater surveillance, the webinar series. The NSF funding for the Research Coordination Network has officially ended, but Ali Baim and I thought we would try and continue the webinar series just to continue the positive momentum that we had from that. So this is the first iteration sort of post NSF funding, and thank you everybody for joining. We have two great speakers that have agreed to speak today. So the first up, actually my PhD advisor, Jordan Peccia, who's the Golden Professor of Environmental Engineering at Yale University. And second up, Ishi Keenum from Michigan Tech. We also have some great speakers lined up for our next webinar, which will be November 7th at the same time. The first one is Fang Chong Ling from WashU in St. Louis, and the second is Aaron Bivens from LSU. So you'll see some emails, reminders about that coming through as well. So with that, I'll stop sharing, and Jordan, you can go ahead and share your screen and get started whenever you're ready.
Thank you, Kyle. Thank you, Ali, for the invitation. So it's my pleasure to talk to you today about our work that I'm entitling this kind of long, clunky title, Daily Wastewater Monitoring as a Tool to Calibrate and Verify Alternative Approaches for Indicating Infectious Disease Outbreaks. The key terms there are daily wastewater monitoring, and also want you to know that this is sort of long-term monitoring, a site that we've been doing for about 5 years. So like many of you, we jumped into wastewater epidemiology and monitoring during COVID. We had worked with pathogens and specifically viruses in wastewater prior to that, but really, the word monitoring crept into our lab right around March 20th of 2020, when the first outbreaks were happening in the United States near New York City. We're about 65 miles away, and we wanted to know what was making its way up to Connecticut. That quickly evolved into a large monitoring effort where we monitored 7 of the largest wastewater treatment plants in the state of Connecticut and worked with the state epidemiologists and CDC to install a wastewater epidemiology program here. It covered about 60% of all the people in the state that were on sewers as well. And, the experience was that it was a lot of monitoring. It was a lot of extractions. It was a lot of reporting, and it quickly took over our lives for a bit of time. We finished that about a year and a half into COVID. And when we were moving on, we started thinking about, well, what do we do next? We'd been so busy working with the state and working as a public service for COVID. How could we turn this into something valuable for research? And we decided just to keep on going with the city of New Haven. So today we are working on samples from day 2023 from the city of New Haven. We haven't sampled every single day, but probably more than 99% of those days. So what do you do when you have 5 years of daily samples of SARS-CoV-2 and other infectious disease agents within one municipality? And how can you leverage that in research? So there's 2 ways that I would like to talk about today. And the first one I'm just going to call service. I think COVID taught a lot of us that there were ways to have impact in the world that went beyond academic sort of continual channels, that we don't always have to give presentations at conferences to have impacts. We don't have to publish papers to have impact. We can have impact where we live. just by the value that's inherent in our work. In this case, we've provided this as a service to the city of New Haven for these last 5 years. So New Haven is one of the cities that can always rapidly look and see what wastewater is doing and understand what's going on with COVID. That was redundant at the beginning, but certainly as testing and information about infectious disease waned after the pandemic, that's become a valuable resource. So the picture I'm showing you here, I'm up in the upper left-hand corner. This is an award that was given to people at Yale University for service to the city of New Haven. The mayor should have been there, but the mayor was sick. This is the president, Mari McGinnis of Yale University. And of course, I'm accepting this award on behalf of my lab. The other thing that we can do with that type of data is research then. And so we've sort of put this into 4 buckets. Initially, we worked a lot because we had data from all these different cities to model and predict case rates and infections from wastewater data. We published studies on that, both case rates prediction and total infection prediction. We did some work on using transcriptome quantification techniques when we were sequencing a lot of genomes of SARS-CoV-2 to try and find the lineage abundances out of those DNA sequences. And then 2 that I'd like to speak about today are DNA sequencing work that we've done for viral metagenomes to indicate the timing and severity of outbreaks for not just a few respiratory diseases, but for a broad variety of infectious diseases. And here we're thinking about the quantitative aspects of that data. And the second one I'll talk about is our use of Google Trends to predict the timing and severity of outbreaks, trying to put at least our lab out of business for daily monitoring. So first, DNA sequencing to indicating the timing and severity of outbreaks. So it looks something like this, and Alessandro Zulli started this work in our lab, and this is a picture of him here below when he was a PhD student. We generally hypothesize that a viral metagenome, this is with a probe hybridization type techniques. We used Illumina techniques, but there are many other, or at least a few other companies out there that are producing these types of kits, that these viral metagenomes can indicate the timing and severity of outbreaks for a variety of infectious disease. What I'm really saying is that it's not just data to identify something, that they can be quantitative enough to do the 2 things that we really care about. That is, what is the timing of the disease and roughly what is the severity of that? And what has enabled us to do some of this type of work is that we've always had the ddPCR monitoring data and the case data to be able to do that. So the graph I'm showing you here is what we called in our lab the quadruple demic. This came right after COVID and after that year where there wasn't much of a flu outbreak. We saw that big RSV pandemic here in green, followed by, not pandemic, rather outbreak, followed by the light blue influenza outbreak, followed by the SARS-CoV-2 outbreak, and in our city, followed by a human metapneumovirus outbreak. We had daily cases for that type of data as well. And so we wanted to sequence those different days, rather those different weeks throughout the cold and flu season to see if we could see that same type of pattern. So kind of just cutting to the chase, this is what the viral metagenome looks like, data looks like. We did this weekly from September all the way through June of 2023. And there's a few things here just by looking at this kind of really coarse heat map that make you think that this might work. The first one is human metapneumovirus, or I'm sorry, this is RSV, orthopneumovirus. You can see that we have high concentrations here during the fall of the year and not much in the late spring and summer. That's what you might expect for that. You can see the same type of pattern in influenza. In fact, the influenza outbreak looks like it happens a little bit after the RSV outbreak, and that's what actually occurred from cases information. Lots of norovirus, a little bit in the fall, but lots more in the late winter, like March. And so if you just do comparisons, these are 4 different plots. One is for RSV, one's for influenza, bottom left is for SARS-CoV-2, bottom right is human metapneumovirus. The blue lines then are going to be ddPCR concentrations, and then the reds are going to be what we got from our probe. These are quantified on the right axis of these plots, and it's reads per million total reads. And you can see that they match up really pretty nicely. The R squareds are at least decent in a lot of these cases. Not so good for human metapneumovirus. I would say that the concentrations were very low in our wastewater. The only time we ever saw peaks was when there was the major outbreak, but it doesn't really capture that curve well. But it does okay for SARS-CoV-2. It does okay for influenza and RSV. And then the power of the type of analysis is that you could just look at the data for all of the different viruses that are revealed in these types of metagenomic analyses. You can see clearly an outbreak of enterovirus A. You can see outbreak types of enterovirus B. You can see rotavirus outbreaks. You can see how common human astrovirus is. You can see little outbreaks of human coronavirus as well. We didn't do anything to smooth this data. And certainly this was weekly data, and if we could get a better time resolved data, it might be expensive and difficult to do, but we could probably get smoother data to do this. There are lots of things I think that we could eventually do to make this data a bit more quantitative. But what we wanted to show here in this study is just that there was clear potential for being able to do this and opens things up from just what you can get from digital droplet or quantitative PCR to a broad suite of pathogens. I would say one final thing about this study, and that is we had a lot of case data for this study as well. And you can see I'm showing you an example here. The top in light blue is influenza. The bottom in green is RSV. The left is wastewater detection of the outbreaks, and the right is the case data. You can see that they match up really nicely in timing and in magnitude as well. And when we're able to do that, we were able to make really nice close comparisons between the cases and between the wastewater data and try to see if there was legs that were present in that data. And we were able to find that the legs actually correlated pretty well with the incubation periods for those diseases. So short legs for influenza that hits you like a truck, and long legs for something like human metapneumovirus and RSV that takes a little bit longer and has a longer documented incubation period. And these are different incubation periods that were mostly reported in the literature. Okay, moving on to the Google Trends data. So this is another thing that we wanted to try, given that we had highly resolved daily data and that we have this data now for, I think, at least 5 different seasons of respiratory disease. So I think we all know the first thing that a person does when they have norovirus infection. And once they get knocked back a bit and they calm down a little bit and leave the bathroom, they might have time to do the second thing, which is they typically Google something. Norovirus, stomach flu, how long for norovirus, how long does the stomach bug take to get over, something like that, right? Well, we can use this deeper and deeper integration between what we Google and having computers with us all the time and our need to find out information on the internet, I think is a proxy to indicate infectious disease. Now, this is something that's been done for quite a while. There were Google Trends searches. There were different types of searches during COVID for things that were seemingly unrelated that revealed that. Some of these are earlier ones with influenza had problems because the case data wasn't strong. But because we had wastewater data now, and we had that stuff daily, and we knew that the reporting if wastewater isn't impacted by how well things are reported or not, we were able to calibrate things and build these models based on our wastewater data. So it looks something like this, and here's our general hypothesis that Google search trends confined to the geographical area that the wastewater catchment is are related to the disease incidence. So on the y-axis here, you have just scaled values. These are values of norovirus wastewater concentrations in green, norovirus cases in red. Case information for norovirus is usually not very good. And then the Google Trend searches, in this case, I'm sorry, the cases are in blue, cases are in red, the wastewater is in blue, and the Google Trend search of just stomach bug is in green. And you can see, while this isn't a perfect match, it definitely goes up during norovirus season. So you could take a lot of different terms. You could think maybe food pipe poisoning might be something they're worried about. Stomach bug, my mother used to call it stomach flu, but apparently stomach bug is more common in the Northeast, nausea, vomiting, rotavirus, or just the term norovirus. And you see different types of correlations. And then, of course, you can just build statistical models based on the strength of these different terms. We built two types of models. One's just a multivariable regression. This is a stepwise regression selection process that I'm showing you on the top. The bottom is the principal components model. Either way, what you can do is use that wastewater data that I'm showing here in the black for norovirus, and you can use that to calibrate a model for stepwise regression based on Google search trends. That's what I'm showing here in blue. And you can see with both of them, you can get very accurate models to match that. You can do the same thing with RSV. In this case, this is the most basic search. We can get a little bit more complicated with it, and we can fit searches to fit this data a little bit better, but I'm just showing you kind of, how simple it can be for something like RSV. Again, the cases is in blue. I'm sorry, the cases are in red, the wastewater data is in blue, and then the Google Trend search here for RSV is in green. It's not a perfect fit, but it's a good indicator of the outbreak in this case. Another simple one is just for influenza A, same thing. These searches go up when the cases go up, as well as when the influenza virus goes up. So it makes me think, because we have been monitoring for a couple thousand days, we hire undergraduates to do this. It takes some time for a postdoc. It's expensive to do as well. And then, of course, it's a constant thing that goes on in your lab to do this monitoring. How much of this we need to continue to do, at least in our case. That is, a long time ago, we needed some way to understand what diseases are circulating in our community. And so the best way to do that was to develop tests. And this is clinical testing programs, and then compiling that test data. Now, something like this will never and should never be replaced, because the individual needs to know if they're sick or what disease they have for treatment, to stay away from people, all of that stuff. But the compiling the test data has been used for epidemiology, and for all the reasons that have been discussed many, many times here in this forum, wastewater epidemiology has some advantages for just indicating community outbreaks in expense, as well as rapidness, as well as the accuracy, because you sometimes just can't cheat wastewater. And what I wonder and want to put into place, at least in our lab, is that search data is ubiquitous, and it is mostly free, and it can be accurate, I think. We've showed in a few cases. And so, what I would like to do then is I would like to continue on monitoring in our lab forever, but instead of only doing wastewater monitoring, I want to gradually, over this year, change all of our monitoring into Google Trends-based models, the monitoring. We can set this up so they can scrape that data, so the models run automatically day to day, and we just put it on sort of run and check in with it week to week to make sure that it's not doing something crazy. We also would continue to validate the data that the models are giving us to make sure that it's correct. But we do have some of that validation already, and I apologize, these slides are a little bit foggy, but this is just something that a new PhD student in my lab, Zoe Zhang, is working on. And she just, in the process of this, so she just sent me the slides really quickly, and we don't have them in the right format to be nice and clear. But anyway, work in progress is what you're looking at is for 4 years of norovirus monitoring in New Haven, Connecticut. Blue is just the model. Everything was calibrated based on this year here, where you can see black, which is the wastewater. We have a little bit of wastewater data for norovirus the next year, but we didn't have anything for this last year. And in fact, it looks right. If you look at the cases, this is what we see. And we happen to know that this norovirus hit early, and there was a big peak in December, which we don't typically see for norovirus. And so it looks like it's performing pretty well. The same thing could be said, I believe, for RSV. This is the same type of graph now. We have more RSV data. This is for the last 3 years, but you can see it captured the outbreak nicely this year. Not so well in the fall of 2023 and early winter 2024, but it captures things nicely again this last year, 24-25. And then finally, this is influenza, and it also captures things nicely in influenza. Again, our model was based just on this year, But if we just extend it out and it's just a comparison between the model and the data now on this year, it fits nicely. It even showed the severity of influenza that was seen across the United States in this last flu season. It'll be more interesting for us to extend this out a little bit more, see what we see here, and also to go backwards and see if we can capture that year where there were many influenza outbreaks based in our model. Okay, so I have just a few conclusions, and I can wrap things up. The first one is that I think that viral metagenome analysis can accurately predict the timing and intensity of respiratory disease outbreaks. That was what we worked with the most, and we had respiratory disease ddPCR to calibrate that. But I think there's a lot there, and I think that is a great tool, not just to identify what viruses are around, but to quantitatively push the envelope here. And then there are directions it needs to go uh, to be able to use that type of data in a way to indicate outbreaks for a large variety of infectious agents. And then the second one is that the wastewater monitoring has some advantages over counting clinical cases, but both are still expensive. And if we really want to democratize this, something like using Google search data that's widely available could allow us to automate this and monitor in an easier way. I would note that Google is commonly used in places all around the world. Of course, it's not just going to be so simple that it works in one area and then you use that model and use it in other areas. I know that there are many other groups, or there are at least other groups that are working on this as well. But I feel optimistic that search terms might be able to replace some of the wastewater monitoring that we do right now. So this is even not a close list of all the people that have involved, but I wanted to show some of the early undergraduate students that worked in our lab: Marcy, Annabelle, Cade, Alessandra Zulli as a PhD student, Zoe Zhang now as a PhD student. Lots of support from municipalities across Connecticut, financial support for the recent work for Jonathan Rothsberg's 4K catalyzer, and also lots of collaborations: CDC, Department of Public Health, School of Public Health, and the Connecticut Egg Station. Thanks, everybody.
Thank you so much, Jordan. That was a great talk. Just a reminder for everyone, any questions you have, there's a Q&A box at the bottom of your Zoom screen. You can enter any questions in there, and you can also upvote other questions that pique your interest. All right, so the first question, Jordan, is for the search trends, how much of that do you think there may be the influence of other data sources or other inputs beyond, an individual being sick? For example, are they more likely to search about RSV when they hear in the news that RSV is on the rise or something like that? Some other sort of signal that you're picking up with the search trends?
It's a good question, and it's the reason why I think things didn't do very well initially when this was tried with influenza several years ago. It's the reason why we haven't even tried it with SARS-CoV-2. It's because there was so much ancillary news coverage about that. But so far, so good. It's worked. I think we depend a little bit on hearing about it in the news because, frankly, a lot of people in the general public aren't going to Google RSV when their child has a respiratory disease unless they know that that's sort of the thing that might be going around. So I think it helps. Interestingly, this was the bird flu year, right? And we thought that this would be a mess, and it worked out nicely. And I think just people having the combination of bird flu and the term bird flu not being in our model helped it work. So far it seemed robust, but it absolutely can get thrown off kilter if there's intense news coverage or some other reason to be Googling some of the terms that end up in our model.
And you mentioned probably the need to refine these models by region. How specific was the trends data that you're able to pull? Was it for Connecticut or a narrower area or a larger area?
It was relatively specific for our city, New Haven, which it was pretty closely covers our wastewater catchment.
Okay. Allie asked an intriguing question. She can't enter it in the Q&A box, but sent me a chat. How do you think this might change as people move towards ChatGPT and other sort of things beyond Google search for these type of questions?
Yeah. Um, I don't know. It's a good question. And we've just started to think about that. Um, I don't know how, I don't know how that's changing Google searches. I mean, Google has its own AI, and so it gives you different responses, but I don't know that the searches have actually changed. And I think that'd be an interesting area for us to look into to see if there are, and this is absolutely true, right? If there are outside sources that are changing the way that people search for information in the first place. But it takes a piece of it for sure.
All right. Here, Caitlin Schaff asks, the search trend model predicts timing of RSV or noro well, but doesn't seem to capture the amplitude of cases. Do you think there's a way to continue to refine that?
Yeah. I think part of the reason is well, if I'm looking at the influenza A here, it doesn't capture these big spikes. And I'm not completely convinced that that's bad. But I think we need to dig more into these spikes. Uh, Interestingly enough, I never thought that we had spikes once we went to ddPCR, but now looking at data like this, it's pretty clear that these are some big spikes. Data here is not normalized in any way. This is not a 7-day average, any of it. And so I think, yeah, uh, it's a good question. I think the really big peaks aren't captured all the time, and we need to figure out whether that is a problem on our wastewater or whether it's a problem on that. Through all of this, problem with the model, through all of this, we have case data as well. And we've done this same type of work for case data. And so if we ever stop, and it works pretty well for case data as well, not things for norovirus, but we have good case data for things like influenza and RSV. And so I guess I need to go back and look and see how well it does with those.
Yeah. All right, I'm going to pool a couple of questions together here. One from Naoko Munakata. How do you think the search trend model will differentiate between similar diseases, right? If people search stomach bug and noro or rotavirus or other things that might go through, or as people respond differently to different diseases in different years, for example.
I think there's going to be some limitation uh, for really closely related diseases. Um, yeah, interestingly, it has worked for things like influenza and RSV when stuff like cold probably should have influenced it. I think we probably see a lot of these spiky things here and there because of small cold, either common cold outbreak or some other respiratory disease outbreak that isn't as widespread. So right now it's looking like, probably more noise than we'd like, but not an overwhelming amount. Rotavirus, I think, tends to be at different times of the year, so we might be able to separate that out as well. But so far, so good. But as we expand it, there's going to be some limitation because symptoms are just going to overlap too much. But I've been surprised how well it's worked with respiratory viruses. So fingers crossed as we move more into things like rotavirus. It's a good question, though.
Yeah. All right, one last question. This was also from Ali in the chat, and I had a similar thought. Can you talk a little bit more about your work between the correlations and the incubation period? And I specifically was interested, what about diseases that might have a much longer incubation period or chronic diseases or things like that? What do you think would be the impact there?
So what type of diseases are you talking about that would have longer incubation period than a week? Like,
Uh, I'm actually not sure. Ali was just asking to talk about it more generally. I was putting in there.
Like Hep A maybe,
Yeah.
or
yeah. Well, to me, I try not to put too much. We thought a lot about what's going on here. We try not to put too much of the mechanism in here because we don't understand everything for sure. But the general mechanism just seems to be right that uh, people are probably shedding a lot more when they're symptomatic. And because they're shedding more when they're symptomatic, They're getting tested more when they're symptomatic, et cetera. We're seeing it in the wastewater, only once that incubation has occurred. I guess is what I'm trying to say there. So, I mean, I don't know if I can read more into it than we can't detect this stuff. To me, it seems like it might be difficult for us to detect things in wastewaters or outbreaks in wastewater until people are having symptoms, if the symptoms and shedding are really closely related. I know that they're not for every disease, but in these types of diseases, they seem to be. That's my best guess. It gets a little too medical for me, to tell you the truth.
Great. Thanks, Jordan. All right, so there were a couple of questions we didn't get to, but I don't know if you'd be willing, Jordan, you can answer those in the Q&A box. And we'll move to Ishi's presentation. So Ishi, feel free to share your screen and get started.
I'm muted. Okay, hopefully now you can hear me and you can see the screen. But someone speak up if you can. All right, so I'm gonna get started today. Hi, my name's Ishi Keenum. Super excited to be here talking to you all today. I'm an environmental engineering professor at Michigan Tech University. And so I'm gonna talk about, I guess, really what I think about when we're doing wastewater surveillance. And this is really kind of the end user, and our goal is to improve public health, and also this need to balance standards and innovation. So I've structured my talk a little bit funny today. We're going to spend some time talking about standards because that's the soapbox I guess I'm going to eternally be on, but I actually like it a lot. And then we're going to talk about, I guess, new targets that my lab's looking at and how we think about this. So the way I like to think about this first is really identifying, and this was really clarifying to me, is our different goals as people conducting wastewater surveillance. We've got kind of the research side and our research goals, and then we've got the goals that the public health agencies have. And I just put this Hughes et al. Table because really a lot of it is trying to understand this relationship between RSV and wastewater, or a target in wastewater, and this statewide positivity or this statewide relationship. And I see myself as a researcher, a lot of my goals are really on identifying new targets, pushing those methods, either it's Jordan using, search terms, or trying to design new assays to look at variant variability, and then trying to quantify and understand the correlation. That's kind of the research side. And we have to figure out, is it working before we want everyone to do it? On the public health side, their goals are different. And I think it's important for us to think about, when we're working with public health labs, what is their end goal, and do they want to spin their wheels on for method development, and what is really a research question we just need to answer. But those goals might be enumerating our disease trends, interpreting those for public health response. That's not a minor thing to do, is figuring out what should we do when we see a trend. And then at different levels, I think if you're at above a local level, you want to understand a little bit of resource management and where you should put your resources for a given population. And obviously, Ali has talked about this at length in this group, but I kind of always envision as this need to compare, the idea that all of our dashboards could be kind of on one mega website, and that we didn't need to look at every different dashboard for every state to then understand trends at a national level or at a full statewide level. And so I still really think about this space for innovation, specifically in the comparison part of what we're doing. And I think a lot of it comes with how we approach monitoring pathogens in wastewater. Basically, as probably everyone on this call at least has heard of, we have a lot of different ways that different labs are doing our processes. And every process that we do is gonna push the needle one way or the other. If you're trying to target viruses, you're gonna want to, select for viruses. If you're trying to target bacteria, you need to select for bacteria. And if you're using like a droplet digital method, those are gonna have different detection limits than, say, a sequencing or an annotation base. But understanding that these different methods exist, and that we want to develop an ecosystem where we can relatively easily characterize the differences in the limits of detection differences or the reach of these methods, I think is important. And so thinking about how, I know bias is kind of a scary word because as scientists, we really want the ground truth of a study, but realizing that when I say bias, it's not always a bad thing. It's really just that it's a decision. And ultimately, to get work done, we have to make decisions. And so all of these little things that we do, though, are going to change maybe the interpretation of our results or might change how comparable our results are. Whether it's just, sample collection, there might be, limitations based on your plant, what you can sample, how you go to concentrate, your extraction methods, what you choose to amplify and how you do it, and then how you choose to handle your controls are really going to all impact the results that you could report on that dashboard and how you're going to communicate with the public or with the public health lab what your results are saying. And so I am just forever going to be saying your lab should use standards. You should use QA/QC standards. And the reason for that is so you selfishly can identify if things are going wrong, but also you can communicate with others what you did, which I think at a minimum, and I know Ali's done a lot with the ME guidelines here, but minimum it's reporting guidelines. And I mean this not just for a research study and a research presentation, but public health labs have public facing dashboards. It's helpful to have a place where methods are talked about. Are these all grab samples? Are these all composite samples? Where does this metadata live? And I understand it can't all be public facing, but keeping a record of it internally for your lab will help you identify changes or things that happened. And often, and I hate that this is a thing, manufacturer decisions can also change your results, specifically on like an extraction bead or a nucleic acid extraction kit. Those are produced in lots. So if you can record what lot that you're using, it might help you identify when you see shifts in your data downstream. And a part of these, so you can also, you have your reporting guidelines, which is basically reporting everything that you do. And I think researchers in research papers, we do this really well, but keeping it going as we are more public facing with our results. But we also have controls. And this is, these are the QA/QC controls that your advisor hopefully is asking you about at every run. I don't know. I have a policy in my lab. We look at those standards results first. And if they don't look good, we don't look at the rest of the plate. Because that's kind of, is the data good? Do we have data to say the data is good? And there's different ways you can do this. And so I like to give people solutions just so you guys can understand what this might mean. Specifically, I think about working it from the outside in. It's a little bit harder to address nucleic acid extraction kind of specifically, but you can at least do your field blanks or your surrogate spike-ins when you're doing method development to really identify, am I getting the target I think I should be getting? And this becomes really helpful if you're trying to, broaden, maybe you initially started concentrating for SARS-CoV-2, but you want to now look for Candida auris. And does your method even capture that? You can maybe use a surrogate to try to see at the beginning, how is this going to go? Am I going to be losing most of what I'm looking for? So you can use it on the method development side, but also on the routine performance side. And I think your different levels of controls will range if you're thinking of a new method versus a routine performance. But with your surrogates, I do also think it's important as we expand our target list to remember that the function of a surrogate that we're using, like a bovine coronavirus or a bovine RSV, might not make sense as you're trying to venture into new types of viruses or new types of bacteria. And so you want your surrogate to match as closely as possible the different characteristics of your target. There are panels you can buy. I know people are using these in this group specifically, but like Zeptometrics makes these different panels that are actually inactivated virus. So this would just be looking at a spike recovery control. But you would know how much you spiked in at the beginning. It could let you get a very, clear recovery of what you're getting as you go. So as you move through your workflow, PCR amplification, I know probably everyone has, I don't know, maybe everyone has run a bad plate on PCR. I certainly have. And it's recognizing that even if it's machines running it, so people make mistakes, but machines also make mistakes, having your controls, even if it's a ddPCR run, are going to be the only way you can tell if your data is good or not. You can look at, the run cycling and your results, but if your field blank is positive, that's a red flag. If your if your target, if you have a control for your target that's just nucleic acids, or you're positive that you spiked in, those relative numbers, you will get a feeling for how they perform over time and what is expected and what is not. But I used to say, only humans make mistakes, but no, we've been using robots and they also make mistakes. So really checking that you're concluding controls on every plate and you're reporting those controls is really important. And these exist, again, you can buy them, Twist makes them. At NIST, we made an MPOC standard that's getting updated and converted to a standard reference material. You can buy them from IDT as a synthetic oligo that you manufacture. In general, I like it to be a longer, messier strain of DNA, but you do what is affordable for your lab. And there are other resources. So just really want to make sure everyone knows that you can buy whole organism cells, particles from ATCC or Microbiologics or Zeptometrics. Like Zeptomatrix, they make them all, and those DNA controls, like I just mentioned. And I think thinking about standards, wastewater surveillance really hasn't had many standards yet. And I will say, it's been a minute, right? So I am a part of the Standards for Wastewater Surveillance Working Group. We have been working with the standards methods for the examination of water and wastewater. We're going to have it's really just a best practices for concentration methods. We're hoping it's going to be submitted in the next couple of weeks here, whenever NIST can open back up. And we'll be ready for balloting, so it should be in the next edition. But I also wanted to let you guys know about resources that are happening more broadly, and maybe this is for public health labs or federal colleagues or state colleagues, but there is another part of wastewater surveillance standards, and I think as we push for wastewater surveillance to become a more integral part of a public health surveillance system in kind of alignment with that clinical testing that will never go away, is we have to start thinking about proficiency testing, and we have to start thinking about how do you ensure or certify that your lab is meeting proficiency standards. This is pretty common in the clinical testing space, but I think in the wastewater surveillance space, it hasn't happened as much. So kind of a first step, Wisconsin has Wisconsin State Lab of Hygiene is a CDC Center of Excellence, and they're running proficiency testing for wastewater surveillance. So they're at least doing quarterly wastewater that they'll mail that they spike in, and you measure it in your lab and report your methods, and you can see, are we generating comparable data? But I think ultimately, as this goes to be kind of further, we're going to need to start requiring proficiency testing of some kind as it gets integrated at larger scales. And so that kind of is the end of those standards part of my talk. I'm going to keep talking about how this is really important for people, because understanding why your data is good or not, I think, is critical. But now I want to pivot a little bit to the kind of the research side, which I will say is a little bit more fun. Well, tick-borne diseases aren't fun, but to me, as a scientist and as a researcher, this is that new target space I talked about. So Michigan Tech, we're located in the upper Midwest, and something that's kind of significantly or an emerging thing here is Lyme disease. And to kind of show you what I mean is really tick-borne and vector-borne diseases that I think we traditionally think of as mosquito-related, but ticks spread over 15 diseases in the United States. I hope, or maybe it's just me, but when I see this little guy on the top here, I don't know if you can see my mouse, it's gross. Everyone hates ticks, which has actually turned to be a really unifying thing. But Lyme disease specifically is really increasing in the US. So in 2005, the case map of Lyme disease looked about like this. We had, a Wisconsin, Minnesota contingent, and then the Northeast. And then in 2023, basically all of Wisconsin's kind of gone if you look at that map, and then the entire Northeast. Lyme disease is really migrating both, north for me in the upper Midwest, but also south along the Appalachian Mountains in the country. And so the different diseases that ticks can spread, they really range, and I'm specifically going to talk about the Upper Peninsula of Michigan, where I'm located, but those 15 diseases might be Lyme disease, which you maybe all heard of because it's the most common one, but it could also be things like anaplasmosis, relapsing fever, ehrlichiosis. There's a whole host of things that these ticks can transmit, and those are the black-legged tick, but then dog ticks also transmit things like tularemia and Rocky Mountain spotted fever, though that's not super prevalent where I live. And so I kind of joined Michigan Tech when we had just received, so as a new PI, you join and you kind of see the landscape of what's going on here. And so Michigan Tech had received an MI Sapphire grant when I got here to really look at the prevalence of tick-borne diseases in Upper Michigan, because Michigan is not considered endemic for Lyme, though Upper Michigan shares a significant land border with Wisconsin. And so one of the things that they did when they were starting this project was look at the prevalence of Lyme disease. You would think in humans, and we'll talk a bit about that case data in a second, but we started with dogs because if anyone like has a pet or has a dog, every time you take your log to the vet, at least I can speak for Maryland, Virginia, and Michigan so far, but I think in many parts of the country, they're going to test them with this IDEX SNAP panel for Lyme or ehrlichiosis and a host of other vector-borne diseases. So our data for dogs is actually really, really good. Just like to say it's much better than people because your dog gets tested every single year and almost all dogs will get tested. And so for the state of Michigan specifically, we saw that, there's about 27,000 CATE positives for dogs every year, which is about 4%. But if we look at Houghton County, which is specifically where we're located, that jumps to about 14%, albeit our case numbers are smaller because we're not a very rural area. But it really started the team thinking about, what's the proportion of Lyme disease carrying ticks in our area. And we'll get to kind of what Jordan was saying about getting into the medical side a little bit as an engineer. But just to kind of talk about this project that happens, because I think it's helpful to understand how this all aligns. There was a big citizen science collection effort at Michigan Tech's campus. This, I guess, is novel because previously, if you do this tick drags to understand disease prevalence, they only got 80 ticks when they were doing this, but throughout this project, we were able to collect over 2,000 ticks. And so it really led us to get a really rich molecular data set for tick-borne disease prevalence. And we had, people enter in the tick and where they found it, and then we did nucleic acid testing for each tick. So when I joined Michigan Tech, we had assays for all of these things working. We were, blending these little ticks and measuring all of their DNA. And they were reporting all of this data on a dashboard. They called it TikTok, but it was really a space where you could get the spatially resolved tick data. And I guess for me as a wastewater surveillance person, I saw this website and I was like, dashboards, where have I seen dashboards? Wastewater-based surveillance loves dashboards. And so I started to think about, okay, we've gotten 2,000 ticks over the past 2 years of sampling. We found that roughly 40% of those ticks carried Lyme disease and kind of decreasing these other diseases. And really probably a naive person like me was starting to think about, can we see this in the wastewater? And I will admit, did not think totally about the shedding dynamics, or I didn't know much about Lyme disease diagnostics. I knew we had nucleic acid methods and I knew we could try. And so, but I want to just start with our challenges up front, because I'm in a town of, our census population is 10,000. It ranges between 7,000 to 14,000, depending on if the school is in session. We have one municipal wastewater treatment plant, but a lot of lagoons. But all of our county is actually incredibly large, and it encompasses many, many, many different treatment systems because we're a rural area with a low population density. To give you just a range, the Western UP Public Health Department, which is a slightly larger scale than our county, covers half of the peninsula of Upper Michigan, half of Upper Michigan. They've just split it into 2 districts. And so when we think about looking at this data, we have to realize that a lot of our epidemiological models that we're working with, they tap out around 10,000 cases or 10,000 individuals. And so we're really at the edge of that. So understanding for wastewater surveillance, a lot of the times we want to compare with clinical trends. We're kind of already set up in a challenging area to do that. And we also know that smaller populations will have just a decreased odds of detection, which is really where things like composite samples might be helpful, but just recognizing statistically, it's not guaranteed to always be detected when you have less contributors. And we also have a lot of people on septic systems. So even if we got, a case reported to the public health authorities, we don't know, is that person contributing to our network? And most people in our county are not on the municipal system. But that being said, we wanted to look at if we could measure and monitor Lyme disease causing bacteria in the wastewater. And so we went about this with digital PCR, initially with grab samples, and then our wastewater treatment plant got a composite sampler, so we switched to composite samples as we could. And looking at Borrelia burgdorferi, which is the causative agent of Lyme disease, with the FlyD gene, but also then just the Borrelia genus, because it can cause other diseases besides just Lyme. And we wanted to make sure, kind of see what trends we saw in our wastewater. So I had an undergrad, Jacob Schmidt, really led this in our lab group, and we've started collecting our wastewater and we started measuring our trends. And I was really following the wastewater surveillance playbook here of, we measure our target, now we've got a year of data, let's see how it looks compared to our public health data. And I think this is where those challenges I talked about really start to come in. And I'm running a little long, but I promise I'll be done very soon. So we started to look at our clinical data. And I think the first thing I want to point out is that our N here, I mean, I don't want people to get diagnosed with Lyme disease, but we only go up to a maximum of 6 per week. So we're talking very, very low numbers. And especially because Michigan is not currently endemic for Lyme, patients don't necessarily get tested very quickly. And also Lyme disease testing is incredibly complicated. It is not something that there's a nose swab for. So the CDC publicly says that we think Lyme is underrepresented here. We thought about looking compared to dog cases, but that website I showed you with all the dog testing that happens, it's really influenced by that rate of positives, but also they only report their data monthly. And so you've got your trends, but it's really hard to say kind of on a weekly basis your correlations. So we've started to do some data analysis here on those trends. I can kind of confidently say we can really confidently detect Borrelia. We can detect those causative agents, but we have not figured out the relationship with cases. And it might be with, companion animals. We have a slightly larger N, but we've got, no statistical power. With humans, we have a very, very spotty data in terms of just the N, like 6 is not that many. So we're still kind of thinking this through and trying to figure it out, but we have been able to sensitively detect a responsive indicator with Lyme disease. And then our next question is really kind of where we all end up with wastewater surveillance, is on where is the Borrelia coming from in our waste stream? And this is where we're starting to work with healthcare providers, because we don't actually have any data on Borrelia shedding, which I think I'm always disappointed at this. We do a lot of Borrelia. If you have Lyme disease and you get a bullseye rash, which most people don't get, that's how we test for Lyme, or it's a blood test, but there's no shedding data or fecal data for Lyme out there. And if you have some, please let me know. But our next goals would really be, can we understand Lyme is one of these that has a really long lag time? We think people are infected for 3 to 6 weeks before they start showing any symptoms. So is it possible that they're shedding in their feces earlier? We don't know. That's a question. And then our question is also on those case data. We know Lyme's underreported, but do people shed Lyme earlier than we think? It's kind of, then can we kind of further go towards community health warnings or location-based data? So I just want to acknowledge, this big Lyme effort was already set up here before I got here. None of this would have been possible if they weren't willing to kind of go with my crazy ideas. Really want to shout out to our Portage Lake Water and Sewer Authority. It's really lovely in rural areas when you go to sample and you tell them you can make them anonymous, and they say, why would we want to be anonymous? We're super excited about this. And so just always asking those things, but people have been really great. And then Michigan Department Health and Human Services and the CDC for this. And yeah, so if you have any questions, feel free to shoot me an email. I love talking about standards and new targets. So excited to hear from you all.
Thank you, Ishi, for that great talk. I'm going to moderate some questions. We have 6 minutes of questions, and there are a couple in the Q&A, but if you have one, please add it so that I can ask Ishi. So I'm going to ask one, which is in the chat. And do you have plans to look for any of the targets for tick-borne illness in the environment? in addition to the work on wastewater, like in environmental waters where there's wild animals, for example?
We are doing some ongoing surface water sampling, so that's pretty easy for us to start to look for. I think we don't have a big study set up. I think we've more been drilling in on the human health side on like where is this coming from and working with people in the state of Wisconsin who's got a pretty robust wastewater surveillance side of things. But we do want to understand where it's coming from. We did look at all of the isolates on NCBI for sources of Lyme disease. It's mostly mice are one of the primary vectors. That's the driver of the tick increased range. So it's possible we'll find it in environmental waters. And we have data, so we should run it on our next plate.
Interesting. Okay. And then this question is a question of mine, but we've been looking a bit at arboviruses, and it seems like one of the similarities in those diseases between the tick-borne illness and arbovirus disease in humans is that it cannot be transmitted human to human. And so humans are kind of like a dead end for the pathogen. And that's sort of been like our working hypothesis as to why we see such low levels of arbovirus nucleic acids in wastewater. And I was just wondering if you had thought about that and how, whether or not you think, wastewater, but it seems like you were able to detect it quite a bit in wastewater. So I was just curious if you had any thoughts about, if you thought about that at all.
Yeah, so I guess we're working under the assumption, and so if we did try to grow Borrelia in the lab, it's incredibly hard to grow in the lab, and it is not transmissible, like it kind of dead ends. It's really that tick, it's a bloodborne kind of transmission, but it doesn't live very well like outside of people or outside of the host. And I don't think it's transmitted human to human, or I haven't seen anyone positing that it is. But it's definitely, we're confused onto what we're detecting, right? We were surprised that it worked. This is why I stuck an undergrad on it to like, really, let's see if this is going to work. And we are getting responsive detections. We're getting them in Wisconsin now too, as we've got more samples from them. And so, yeah, we're trying to understand what's going on here. Yeah.
And I guess another question that just came up in the Q&A is it possible to test for antibodies of Lyme disease in wastewater? I mean, that would require a completely different set of methods that you probably don't work on. So
I think that's a really cool idea. I am totally up for trying it. But we haven't explored that so much. I think we've been really focused on trying to get whole genomes out of wastewater and trying to compare those genomes to Borrelia isolates on NCBI, but that is because I am an omics person, and so to me that was a very tractable next step. If anyone, I just don't know anything about antibodies, but it sounds like a great idea.
Okay, well, good luck with the tick-borne work. It seems really important and interesting, and a lot of challenges working with bacteria as opposed to viruses, I think, because they can be so similar to close relatives and not be the target. So there's a question about the standards part of your talk that I wanted to ask, and if anyone has any others, please add them. Thanks for being such a big proponent for reporting and standards, and you would think that that our field would be really good at all of that, and it's surprising how actually there's a lot of missing information, and part of it is probably because people just take for granted that, oh, you just know, of course I did a control, I don't have to tell you. But the question here is like, are you thinking about like synthetic wastewater matrices for validation and proficiency testing? And how could that be done when the wastewater matrix is basically like an undefined, like infinitely complex and changing matrix? Like, what are your thoughts on that?
Oh yeah. So NIST is working on this. Let me, I guess, just say I started working on this at NIST. They are working on QA/QCing the stability and like how you could store such a synthetic wastewater over time. So we're working on it. And it's really the idea, I guess, because every wastewater is different, your synthetic wastewater is not going to mimic any real wastewater 100%. I think it's just accepting that. but it provides a really good way. And what we've been working on is making sure there are hit inhibitors. So it isn't gonna extract really nicely like a water sample. But it's going to have some inhibition and it's going to have some solids fraction. And so that you can do these different methods and you could try to spike in. A lot of the work at NIST is then thinking about homogeneity and stability of that material, what it would look like. Is it a documentary reference and storage? But it's not, yeah, I think when I started, I was just going to solve synthetic wastewater. I think if you guys are noticing a trend there, I get excited about things. But they are working on the homogeneity stability. I'm hoping that will come out soon. But I'll check in with them on that. But there is, I guess, for recommendations, we were finding there's a cat food method that is used a lot in bioreactor circulation. And you don't want it sterile, right? You don't want your synthetic wastewater to be sterile because wastewater is definitely not sterile. So the cat food is also not sterile, but that was part of getting it to consistently blend. Everyone has to have the same blender. So we're working on kind of those additional method challenges.
I don't really want to work with cat food myself, but anyway, thank
It's the
you.
little pellets, it's kibbles.
Thank you so much for the talk. We're at time, so we're going to end the webinar. Thank you everybody for joining, and we'll see you next month. Thank you. Thanks, Jordan. Thanks, Ishi.
Bye.
Bye.